Assalamu alaikum, everyone. My name is Rayan Ahmed. I'm a second year medical student, and today I will be giving you a PAL session about two lectures combined into one. So, the first lecture I will be giving you a PAL session about is the pentose phosphate pathway and the alternative pathway of carbohydrate metabolism. And before I begin, I know this lecture is hard for some of you guys, and some of you guys might find it easy, but inshallah, after this lecture, after this PAL session, it's going to be very easy. And feel free to interrupt me throughout the session, ask me questions, and here's my contact information. You guys can text me anytime, and with any difficult questions, and I'll be there for you guys. Okay. Let's begin. So, first we're going to be talking about the pentose phosphate pathway. So we have four objectives. The first one is three pathways that utilize glucose in cells and two products of pentose phosphate pathway. The second objective is the biochemistry of the pentose phosphate pathway. The third objective is transketolase in pentose phosphate pathway and Wernicke-Korsakoff syndrome. The fourth objective, which is three uses of NADPH in liver, immune cells, RBCs, and hemolytic anemia on G6PD deficiency. Guys, I want you to keep in mind the three objectives that I just talked about is very high yield, and I need you guys to focus in most of them. Good. Let's begin. So, three pathways that utilize glucose inside cells. So, we already know about the glycolysis pathway, which produce ATP, and then we also know about the gluconeogenesis pathway, which glycogen storage right here, which use ATP to produce glycogen, right? We also have another pathway, which is the pentose phosphate pathway, which does not use ATP or gain ATP. It doesn't have to do anything with ATP. Glycolysis, we already know the main purpose is to produce ATP, and the glycogenesis, we have glycogen storage. Again, it uses ATP to produce glycogen storage. And then when we don't want to use any ATP, we have the pentose phosphate pathway. Again, so what's the purpose of pentose phosphate pathway? There are two main purposes of pentose phosphate pathway, and that's it. I just need you guys to remember two simple products. Again, what is it? NADPH and ribose. It has a full name, but I just need you guys to keep in mind ribose. So we have NADPH and ribose. Okay. Now, pentose phosphate pathway. You guys are already familiar with this pathway, which is the glycolysis pathway. Again, the main function of glycolysis pathway is to produce ATP and pyruvate, of course. So, pentose phosphate pathway, also known as hexose monophosphate shunt, which is a name I need you guys to remember. And then we also have 6-phosphogluconate pathway, less common name. I highly doubt you guys have to remember this name, but just keep it in mind. Again, hexose monophosphate shunt. Why is it called a shunt? Because it does not take the usual pathway of glycolysis. Rather, it shunts to the side as it hexose monophosphate shunt. Again, hexose because glucose is a hexose as well. And monophosphate as well here, monophosphate. And I need you guys to remember this one fact. It says, occurs in the cytosol of all cells. Pentose phosphate pathway is always occurring in every cytoplasm of all cells. Okay. And the aim of pentose phosphate pathway is to give important products, not energy. So ATP is not included. Again, remember, I told you guys this. ATP is not used or gained. It is gained in glycolysis and glycogen storage. It is used. In pentose phosphate pathway, no ATP is used. I need you guys to remember that. Okay, let's move on. Now, pentose phosphate pathway. Again, we're gonna be revisiting concepts throughout the lecture, so I need you guys to link stuff. The purpose of pentose phosphate pathway was to synthesize NADPH and ribose 5-phosphate. NADPH has many uses. I need you guys to only know three important ones, which we're going to be discussing later on. And ribose 5-phosphate is to synthesize nucleotides. I need you guys to know this. Ribose 5-phosphate, which is a product of PPP, is used to synthesize nucleotides. Now, pentose phosphate pathway is, let me move this. Yeah, pentose phosphate pathway consists of two phases. So we have the oxidative phase right here, and then we have the non-oxidative phase right here. In the oxidative phase, it's called irreversible. As soon as you get to ribose 5-phosphate, it can't go back to glucose 6-phosphate. That's irreversible. And the rate-limiting enzyme, which is so high yield, they're going to ask you about it. It's glucose 6-phosphate dehydrogenase. It's used right here to produce an NADPH and to produce ribose 5-phosphate. I need you guys to remember this. Anything in red, by the way, you guys need to remember this. Okay. glucose 6-phosphate dehydrogenase, G6PD, the rate-limiting enzyme of the oxidative phase in pentose phosphate pathway. Very important. The second phase is the non-oxidative phase, which is reductive phase. A series of reversible reactions resulting in interconversion of sugar in a molecule. As you guys can see, there's arrows in both directions. That indicates that it's a reversible reaction. And ribose 5-phosphate is one of the products of pentose phosphate pathway and NADPH. I need you guys to also remember ribose 5-phosphate and NADPH again. And remember, in pentose phosphate pathway, we have no loss or gain of ATP in this process. It's not like glycolysis. It's not like glycogen storage. Okay, let's move on. Now, we're going to be moving into the oxidative reactions in pentose phosphate pathway. So we start from glucose, okay, and then we convert it into glucose 6-phosphate. You guys already know this through glycolysis. But it takes a shunt, which is the hexose monophosphate shunt. In this shunt, the rate-limiting enzyme is glucose 6-phosphate dehydrogenase, G6PD. And in this pathway, NADPH is being produced 2 times, as you guys can see here, to make ribose 5-phosphate. And this is the rate-limiting enzyme, which is, again, G6PD, glucose 6-phosphate dehydrogenase, and 2 NADPH are produced in this process. I need you guys to remember 2 NADPH and glucose 6-phosphate dehydrogenase from this oxidative reaction in pentose phosphate pathway. And again, remember, this is an irreversible reaction, not like reductive reactions, which is going to be the next part right here. So reductive reactions, this is the second part. And the reductive reactions, it starts from ribose 5-phosphate, which is a product of pentose phosphate pathway. It goes back into glycolysis by this enzyme called transketolase into fructose 6-phosphate. So transketolase is the enzyme that I need you guys to remember in reductive reactions. It is very essential for normal brain function. Please keep this in mind because we're going to revisit this concept. Essential for normal brain function, transketolase. Okay, let's move on. Now, we're going to be talking about feedback inhibition of NADPH. So, as you guys can see, we already talked about this. This is glucose 6-phosphate, 6-phosphogluconate. This is the oxidative reaction. And the enzyme that is being used here is G6PD. So the levels of NADPH regulate G6PD. Therefore, it regulates whether glucose 6-phosphate right here goes into pentose phosphate pathway or into glycolysis. So now the products that we had was ribose 5-phosphate, which was pentose phosphates. And then one of the other products we had was NADPH, right? When NADPH levels increase, there's a feedback inhibition of G6PD towards the glycolysis pathway rather than the shunt. So what I mean by here is when NADPH is increased, we have a lot of NADPH. We don't need more NADPH being produced in the pentose phosphate pathway. This will go inhibit the rate-limiting enzyme, which is glucose 6-phosphate dehydrogenase, to tell it to stop making more NADPH. And rather than finishing the pentose phosphate pathway, it's going to go back into glycolysis to produce more ATP. It's like a feedback inhibition. You guys already know this from previous MOL lectures, but just keep this in mind that NADPH inhibits G6PD when there's too much. And yeah, that's about it. Okay, let's move on. Now we're going to combine glycolysis and pentose phosphate pathway. And when you guys look at a picture like this, I know you guys get overwhelmed or it might look a little scary, but inshallah, it'll be easy because we're going to be breaking this down into simpler terms. Okay. So remember when I said the products of pentose phosphate pathway, there was ribose 5-phosphate and NADPH. We already talked about NADPH being a blocker of pentose phosphate pathway. Now we're going to be talking about ribose 5-phosphate being a blocker. So when ribose 5-phosphate is increased right here, transketolase, which was the enzyme in the reductive phase, and transaldolase, you guys don't need to know about transaldolase, but just keep it in mind. Now, transketolase and transaldolase produce glycolytic 1 intermediates, which are these. You guys don't need to know this. I put an X right here. You guys don't need to know these intermediates. All you guys need to know is that ribose 5-phosphate, through the help of transketolase, TPP, can go back into fructose 6-phosphate and glyceraldehyde 3-phosphate for ATP generation for glycolysis. Again, when there's a lot of ribose 5-phosphate, we don't want more. We can block the whole pentose phosphate pathway by the help of transketolase, making more of glycolytic 1 intermediates, which are fructose 6-phosphate and glyceraldehyde 3-phosphate into pyruvate for ATP generation. So let's go over it again. We're going to be speaking about, again, NADPH being a blocker and ribose 5-phosphate being a blocker. When ribose 5-phosphate is a blocker, it blocks the pentose phosphate pathway by using an enzyme called transketolase. Transketolase helps ribose 5-phosphate convert into the glycolysis intermediates right here, and then it goes into pyruvate and produces ATP. When there's a lot of ribose 5-phosphate, again, it's end product inhibition. Okay, continuation. Deficiency in ribose 5-phosphate, more pentose phosphate pathway. Again, remember when we said when there's a lot of ribose 5-phosphate pathway, we're going to block the pentose phosphate pathway and we're going to go into glycolysis. That's when there's no ribose 5-phosphate, we're going to use more PPP. From now on, I'm going to be using PPP, not pentose phosphate pathway, because it's very long term. Anyways, so ribose 5-phosphate goes into glycolysis again when there's a lot. That's when there's barely any. When there's a deficiency in ribose 5-phosphate, we're going to have more pentose phosphate pathway occurring. When ribose 5-phosphate demand increases, the non-oxidative pathway, which is the reductive pathway, functions in reverse. Transketolase, remember, I need you guys to remember this enzyme, transketolase, and transaldolase catalyze the conversion of fructose phosphate and glyceraldehyde 3-phosphate into ribose 5-phosphate. So when we don't have a lot of ribose 5-phosphate, transketolase, this enzyme, which was right here, is going to use fructose 6-phosphate and glyceraldehyde 3-phosphate back into ribose 5-phosphate, because this is used for a number of reasons to make more nucleotides. We're going to be discussing this later on. So I hope you guys understand that. And if you guys don't, please feel free to ask questions. Okay, let's move on. Again, transketolase, guys. It requires TPP, which is thiamine pyrophosphate, as cofactor. You guys know this from previous lectures as well, but just add another enzyme that it requires transketolase as well. And TPP is the active form of thiamine, which is vitamin B1. Please keep this in mind. They might ask you guys multiple choice about this as well. So TPP is the active form of thiamine, pentose, ribose 5-phosphate to glycolytic intermediates without going through G6PD reaction. Remember, guys, the whole point of transketolase is to convert pentose, which is ribose 5-phosphate, to glycolytic intermediates without going through G6PD reaction. We can go back. I can show you guys right here. The whole point is to skip this whole pathway and go again into fructose 6 pathway and glyceraldehyde 3 pathway in glycolysis. We don't want to use G6PD. We can go straight directly by using the help of transketolase from ribose 5-phosphate into fructose 6-phosphate and glyceraldehyde 3-phosphate to make ATP in glycolysis. And there are other enzymes that you guys must know that require TPP as cofactors that you must remember. You guys can also use the lovely coenzyme for nerds, transketolase, PDH, these two enzymes. You guys need to know this. But remember, transketolase, it needs vitamin B1, which is thiamine, and TPP, thiamine pyrophosphate, as cofactors. Okay, let's move on. This syndrome is very important. I'm pretty sure they're going to ask you a question about it. Wernicke-Korsakoff syndrome, caused by a severe lack of thiamine, vitamin B1, which was what? TPP. TPP is the active form of thiamine, vitamin B1. Without TPP, transketolase cannot function. Transketolase requires TPP. So when we don't have thiamine, which is vitamin B1, which is TPP, we're gonna have a syndrome called Wernicke-Korsakoff syndrome. Most commonly seen in alcoholics. Other causes could be malnourished, malabsorption, etc. But the most important one that you guys need to know is alcoholics. Whenever you guys see severe lack of thiamine, vitamin B1, and that person is an alcoholic, just think Wernicke-Korsakoff syndrome. And transketolase has reduced affinity for TPP in Wernicke-Korsakoff syndrome. So what that means is transketolase requires vitamin B1, which is thiamine, right? TPP. It has reduced affinity for TPP in Wernicke-Korsakoff syndrome. Now, symptoms, you guys need to remember this: confusion, inability to coordinate voluntary movements, which is actually, and eye problems. And when these symptoms are prolonged, they can cause lasting brain and nerve cell damage, leading to severe memory loss. Remember, thiamine was essential for normal brain function. I said this in the beginning. Thiamine was essential for normal brain function. So look, TPP is the active form of thiamine vitamin B1. Transcrystallase needs TPP. What happens in Wernicke-Korsakoff syndrome? The transcrystallase has reduced affinity for TPP in Wernicke-Korsakoff. And they might give you a scenario where a person's confused, cannot move, alcoholic as well, eye problems, and brain problems as well. Just think Wernicke-Korsakoff syndrome. And they must say that severe lack of thiamine, vitamin B1. Okay. Let's move on. Now, checkpoint. Any questions? If you guys have any questions for me, please ask. If you guys don't have any questions, I have a question. Which of the following statements about the oxidative phase of the pentose phosphate pathway is correct? It's being recorded anyway, so I will say that. It's it is B. The PPP supplies ribose 5-phosphate and NADPH in the quantities the cells require. Okay, amazing. I can explain it as well. Which of the following statements about the oxidative phase of the pentose phosphate pathway is correct? The rate limiting enzyme reaction of the PPP is no, it's not. It's not phosphatase. It's a glucose 6 phosphate dehydrogenase, G6PD. The PPP supplies ribose 5-phosphate and NADPH in the quantity that cells require. It's quite straightforward. That is the correct answer. C is incorrect. No, we make NADPH. We don't make NAD plus. And again, D is also incorrect because we generate NADPH, not NADH. Okay, let's move on. Another question for you guys: which enzyme is needed as a cofactor for transcrystallase enzyme? Thiamine, yes, it is. Requires TPP, thiamine pyrophosphate as a cofactor. Amazing. Okay, let's move on. Okay, so now we're going to be doing the products of the pentose phosphate pathway. Remember, I said two products that I need you guys to keep in mind. It was ribose 5-phosphate and NADPH. NADPH is similar structure to NADH. Please don't get confused between NADH and NADPH. Whenever you think NADPH, please remember the pentose phosphate pathway. Remember all the rate-limiting enzymes. The main one for this one is going to be G6PD, glucose 6-phosphate dehydrogenase. And NADH is used for oxidative phosphorylation. You guys need to remember that. But for NADPH, please remember PPP. NADH is used in multiple areas. One of them could be used for oxidative phosphorylation. And now we have the ribose 5-phosphate. Now, ribose 5-phosphate is important because, it is a component of nucleic acid. So we have purine nucleotides and then pyrimidine nucleotides. You have adenosine, guanosine. You guys already know this. Just remember, ribose 5-phosphate is needed for nucleotide synthesis right here. That's all you guys need to know. Okay, let's move on. Now, remember what I said that you guys need to remember 3 functions of NADPH in cells? Well, these are it. So we have number 1, reductive biosynthesis, anabolic pathways, cofactor and fatty acid and steroid synthesis, such as the liver, mammary glands, testes, and adrenal cortex. The second one I need you guys to remember is bactericidal activity in neutrophils and phagocytic cells. And the last one we have is protection from oxidative damage, such as in RBCs. Okay, I know it might feel a lot, but it's going to be very easy, inshallah. Okay, the first one, reductive biosynthesis. NADPH in the liver, very important. The first one is reductive biosynthesis. NADPH in liver, it synthesizes fatty acids, cholesterol, and nucleotides. And they're always made in the liver. They're all made in the liver. I need you guys to remember this. So NADPH is used in the liver to make fatty acids and cholesterol. That's all from the first point, which is reductive biosynthesis. Remember, we had this is the rate-limiting enzyme in the oxidative phase, and then we have NADPH, 2 of them being produced to be precise. And then biosynthesis was fatty acids, cholesterol, and nucleotides. right here in the liver. Okay, let's move on. Now, we have bactericidal action of NADPH, also known as respiratory burst. It's oxygen-dependent killing. So we have neutrophils, phagocytes, whatever. NADPH basically generate hydrogen peroxide, which is H2O2, to kill bacteria. Look at here, this is the whole pathway. So we have oxygen, normal oxygen, and neutrophils, phagocytes, whatever. NADPH comes by using the enzyme NADPH oxidase. Remember that, please, to make oxygen radical right here. And then they use another enzyme, superoxide dismutase, which is hydrogen peroxide, to make, again, bacterial death. And then myeloperoxidase, by using the help of the chloride ion to make HOCl, to bacterial death as well. All these free radicals and through the help of O2, they all kill bacteria. This is the whole point of respiratory burst, and it's oxygen dependent killing. Oxygen dependent because without oxygen it wouldn't work. We need an oxygen as well here. And all these pictures are from your slides, so don't worry about that. And I need you guys to remember these enzymes, please. They might give you any enzyme, NADPase oxidase, superoxide dismutase, myeloperoxidase. Just keep any of them in your mind, or keep all of them. That would be better. And the last one, function of NADPH and RBCs. This is a very important one because there's a deficiency of one of the concepts we're going to be talking about, and I need you guys to remember that as well. So NADPH is very important here in RBCs as it needs to convert glutathione disulfide into glutathione by the enzyme glutathione reductase. A lot of enzymes, a lot of names. Let's break it down. Okay. So in RBCs, we have glutathione disulfide right here, normally present in red blood cells. NADPH comes from the PPP or HMP shunt and makes the glutathione through the enzyme glutathione reductase. Okay, and glutathione can be converted into hydrogen peroxide. Can convert hydrogen peroxide right here into H2O through the enzyme glutathione peroxidase, which is non-toxic. Okay, this H2O2 hydrogen peroxide is toxic. H2O, which is normal water, is not toxic. Now, H2O2 is a reactive species which can create oxidative stress in the cell and by free radicals right here and kill bacterial death. So this can lead to the death of RBCs. But we have glutathione, which is produced by the help of NADPH through the enzyme glutathione reductase from glutathione disulfide. Sorry. There's a lot of names, but just remember glutathione and glutathione reductase. These are the two that they are going to most probably ask about. So glutathione is needed to convert this toxic hydrogen peroxide into non-toxic right here by the enzyme glutathione peroxidase, okay? Through the help of, again, where it all started, NADPH. So we don't want a lot of H2O2 hydrogen peroxide in our red blood cells because this is going to cause oxidative stress and they're going to cause free radicals and it's going to kill the red blood cell. So we don't want this. We're going to convert this into H2O2. But what happens when we don't have NADPH or we don't have this enzyme or we don't have glutathione? This is going to accumulate, right? That's all. Moving on to the next slide. Now, very, very, very high yield. This it's going to be very easy, inshallah. So G6PD deficiency. We know that G6PD is the rate limiting enzyme in the whole pentose phosphate pathway, which is used in the oxidative part of the pentose phosphate pathway. Now, if we don't have G6PD, we're not going to have NADPH, right? So deficiency of NADPH leads to decreased amount of reduced glutathione. Basically, it's enough to deal with normal amounts of H2O2, and individuals are usually asymptomatic. But whenever there's a trigger, a lot of H2O, a lot of hydrogen peroxide exceeds the reduced glutathione, leading to destruction of RBCs, hemolytic anemia. That's what happens. So again, let me just explain it in this pathway. What I mean by, let's say we don't have NADPH, right? We don't have G6PD. We don't have G6PD. If we don't have G6PD, we're not going to have NADPH, right? So when we don't have NADPH, the reduced glutathione, which is this one right here, but through the help of glutathione reductase and glutathione disulfide, we're not going to have a lot of reduced glutathione, which can convert hydrogen peroxide into normal H2O. If we don't have a lot of this is going to accumulate, and then it's going to lead to oxidative stress and in the cell by free radicals and kills the RBCs. But if the thing is we um uh, it's normal to have it's I mentioned it over here. It's enough to deal with normal amounts of H2O2, right? If we don't have a lot of NADPH. But whenever there's a trigger, H2O2 increases. A lot of H2O2, hydrogen peroxide increases, and reduced glutathione can't deal with it. So this leads to bacterial red blood cell death, right? And it causes hemolytic anemia. Hemolytic anemia. Remember, I'll need you guys to link two definitions: G6PD deficiency into hemolytic anemia, right here. And the three triggers that I talked about, and there's a presence of a trigger, the amount of H2O2 exceeds. There's three triggers that you must know. First of all, they're fava beans right here, like green fava beans. It contains oxidants. Favism is an acute hemolytic anemia that is seen in G6PD deficient patients after ingesting fava beans. They might give you guys a case scenario, where a person has ingested fava beans and has G6PD deficiency. So I need you guys to remember this very important trigger. Now, we also have infections that lead macrophages to generate free radicals. Sorry, right here. Free radicals that are being generated in macrophages and neutrophils. This could be another trigger. And we have drugs. We have antibiotics, antimalarials, aspirin, acetaminophen. You guys don't need to know what type of drugs. It's all for you. You guys don't need to know this, but just keep in mind drugs such as antibiotic, antimalarials, and aspirin. Okay, and acetaminophen. No, all three triggers increase the amount of free radicals, which is H2O2. When there's no trigger, the decreased amount of reduced glutathione is enough to deal with normal amounts of H2O2. But these three triggers lead to excess amount of H2O2 that exceeds the reduced glutathione, leading to destruction of RBCs, hemolytic anemia. Please remember this. Any questions here? Okay. Let's move on. Okay. Now, G6PD deficiency, most common human enzyme disorder. We also have X-linked disorder, MILS. Just keep these 2 points in mind: most common human enzyme disorder and X-linked disorder in males, most common in males. High prevalence in Africa, Asia, and may protect against malaria. We don't know why, it just may protect against malaria. Now, we also have clinical features, which is dark urine, jaundice, anemia. Anemia, we have fatigue, paler, whatever. I can show you guys. Jaundice, remember yellow, sir, and then paler. Just remember, clinical features of G6PD deficiency, dark urine, jaundice, and anemia. Hemolytic anemia, right? And again, they might give you a case scenario that you guys need to know that the person has dark urine, jaundice, again, anemia, hemolytic anemia. Now, the fact that it's anemia, how will it look like? Right here. Classic finding of G6PD deficiency. And again, this leads to hemolytic anemia, right? We also have Heinz bodies, which is oxidized hemoglobin precipitated in RBCs. Little dots you're going to see in RBCs right here. Like, just like that, keep that in mind. One or few dots in the red blood cells, RBCs. Heinz, we call them Heinz bodies, okay? Oxidized hemoglobin precipitated on RBCs. Okay, and then we have bite cells. You guys might be familiar with this from FND, but I'll just say it again. Phagocytic removal of the membrane by splenic macrophages. If you guys can't remember how bite cells look like, they look like Pac-Man. So, I mean, yeah. That's about it. Remember, there's Heinz bodies and bite cells in G6PD deficiency. So if they give you a case scenario, they might give you dark urine, jaundice, anemia, and then they're going to describe the anemia. They're going to say Heinz bodies we're seeing and bite cells we're seeing. Just think that it is hemolytic anemia. And why is it? Because of G6PD deficiency. Okay. I know this was a lot. If you guys have any questions, please let me know, because we're about to finish with the first lecture. Okay. And this was mentioned in your slides, but I highly doubt they're going to ask you a question about this, but we'll go over it again for completion's sake. So it is used for G6PD deficiency diagnosis. What is used? Fluorescent spot test, FST. if we want to diagnose G6PD deficiency, we can use fluorescent spot test, FST. Now, how does it being used? It's basically natural fluorescence of NADPH under ultraviolet light. So we have natural fluorescence of NADPH under ultraviolet light. Add glucose 6 phosphate, where it all starts. You guys remember for pentose phosphate pathway, we have glucose 6 phosphate and NADP. Now, through the natural fluorescence, we're going to measure NADPH fluorescence. Normal G6PD activity produces fluorescent NADPH. You're going to see like fluorescent out of NADP. While in G6PD deficiency, you're not going to see any fluorescence. And it's basically used for screening and diagnosing newborns. So again, we basically use natural fluorescence of NADPH under ultraviolet light. We add glucose 6 phosphate, right here, glucose 6 phosphate and NADP to measure NADPH fluorescence. If the person is not deficient of G6PD, it's going to produce normal NADPH. But if it is, there no fluorescence in seed. Okay, that's about it for this. Very low yield, but again, completion's sake. This is the summary of G6PD deficiency. We have mutations of DNA of G6PD gene. I don't know why we don't have G6PD being produced. Rate limiting enzyme is gone. No NADPH. Enzyme activity decreases. Production of NADPH decreases as well. Reduce glutathione. Very important. Reduce glutathione decreases. Reactive oxygen intermediate, which is hydrogen peroxide, increases. Damaging to red blood cell wall. Hemolysis, hemolytic anemia. Hemolytic anemia is the most important thing you need to know from G6PD deficiency. Okay. And that's about it. Okay. You guys have any questions? Please let me know. If you guys don't, I have a question for you guys. Okay. So here's a question. We have a 25-year-old African-American male in good health had read about fava beans in silence of the lambs for dinner one night. The man had liver with fava beans. Okay, yeah, you guys. Okay, you answered it already. It is C. Yeah, good work. Okay, I don't even need to explain it because you guys got it right. Yeah, reduced glutathione. Yeah, it is. If you guys need me to explain it, I can explain it as well. Showed hemolytic anemia. The patient has most likely a defect in regenerating which of the following? Let's go back. G6PD deficiency, we don't have any DPH being produced. Again, so we need glutathione disulfide, we need glutathione reductase, but we don't have glutathione, which basically converts the trigger into normal. So if we don't have reduced glutathione, that's the problem in regenerating. Simple as it gets. Okay, nice. And we're done with the first lecture. We're going to be moving on to the second lecture right here, but I'll just give a 2-minute break and then we're going to start again. So. Okay, this is the second lecture of today's class session. We have alternative pathways of carbohydrate metabolism. And this lecture is very dense, so please focus with me. Okay, we have 3 objectives. Literally 3 objectives, no more than that. I know in your doctor's slides, you guys have a lot of lectures, a lot of objectives, but I split it into 3, okay? We have all about fructose, all about galactose, and then all about lactose, right? We're going to be starting off with fructose, going into galactose, then going into lactose. Galactose, lactose sound similar, but this is different than this. Fructose is the main high yield objective of this lecture. I think like 70% of this whole lecture is fructose, so please focus with me. Okay, let's begin. You open your doctor's slides, you're going to look at this huge picture that he has put. And honestly, no one can understand that. Not even I can understand that. I don't know how he expects people to understand every single thing here, but you're going to be basically learning every single thing here, but like broken down into simpler terms. So please don't get overwhelmed whenever you open your slides and look at big pictures such as this, or even this, because it gets very simple, okay? So let's begin. Before we get into the objectives, we need to know about building blocks. So you guys know monosaccharides. You guys know about glucose. You guys know about fructose, galactose, and glucose again. So glucose plus fructose, monosaccharide will give you what? A disaccharide, right? Two sugars combined together. We have sucrose. Please keep this in mind. Glucose plus fructose equals sucrose. Another monosaccharide, which is glucose, another monosaccharide, which is galactose, gives you lactose. Another glucose and another glucose together combined gives you maltose. Glucose plus fructose gives you sucrose. Glucose plus galactose gives you lactose. These two are very important. Maltose isn't that important, but just keep it in your mind. But sucrose and lactose, please, please, please don't forget it. Because we're going to be building based upon literally these two definitions. Okay. Overview. We have all these saccharides that contribute to energy metabolism. We know that glucose already does monosaccharides, such as, and then we also have more monosaccharides that contribute to energy metabolism. So we have fructose, galactose. These two monosaccharides also contribute to energy. And then both of them can enter glycolytic pathway. As you guys can see, like there's a little flow that glucose is the main thing. Everything leads back to glucose. So fructose and galactose can also lead to glucose, some one way or another. You guys just need to find out how. It could be through the glycolytic pathway, through its own pathway. We're going to discuss. Again, remember, glucose plus glucose gives you maltose. Glucose plus fructose gives you sucrose. Glucose plus galactose gives you lactose. And for all these disaccharide sources, it could be milks, fruit, sugar cane, just, yeah. Okay, let's move on. So now we're going to be getting into our first objective, which is everything about fructose. Okay. Okay, let's begin. So now, a little background about fructose. The major source of fructose, remember, fructose is a monosaccharide. Major source of fructose is the cleavage of sucrose, which is a disaccharide. So when we break down sucrose, we're going to get glucose and fructose. Okay. Again, breaking down sucrose into glucose and fructose that's the whole point, literally. You guys need to remember cleavage of sucrose gives you glucose and fructose. And now, fructose can also be obtained from fruits. That's very obvious. Fructose, fruits, same thing. Entry of fructose from blood to cells is not insulin dependent. This is so important because the past we have learning, not past lectures, not this lecture, but like glycolysis, whatever. The entry of glucose from blood to cells is insulin dependent, but fructose isn't. That is so important because this is going to help us in a lot of pathways for fructose. Remember, entry of fructose from blood to cells is not insulin dependent. It does not need insulin. It does not need insulin. And fructose does not promote the secretion of insulin, as we just said, not insulin dependent, so is glucose. Remember, glucose, insulin, they both counteract and they kind of need each other. But fructose is very independent. It does not need insulin at all. Okay, let's move on. Okay, so we're going to be talking about how fructose is made. Okay, how is fructose made? So we have glucose. We already know glucose. Everyone knows glucose. Glucose is converted into sorbitol. Please remember sorbitol. This is a new name for you guys. So remember sorbitol. Sorbitol is then converted into fructose. Now, there are two enzymes. Please remember them as well. I know there's a lot of enzymes, but once you do them like twice, you're going to know the enzymes very well. So the first enzyme that I need you guys to remember is aldolase reductase. Aldolase reductase is used from glucose into sorbitol. Okay? Glucose into sorbitol, aldolase reductase. And then we have from sorbitol into fructose, it's in the name, sorbitol dehydrogenase. Breaks down dehydrogenase, sorbitol into fructose. Sorbitol is a new name again, please remember this. This pathway, the fructose being made, is very critical in seminal vesicles, as fructose provides sperms with nutrients. This is very, very important. I'm pretty sure they're going to get you guys a question about this, so this slide is it. Fructose basically provides sperms with nutrients. And if you don't have fructose being made, fructose deficiency, male infertility. Remember the enzymes, aldolase reductase, and sorbitol dehydrogenase. So if we don't have fructose being made, male infertility, because sperms are not getting nutrients and they cannot move. So you're going to have male infertility. Okay. That's about it. Enzymes and sorbitol is the new name that you guys must know. Okay, let's begin. Now, now we have sorbitol and hyperglycemia. Okay, So we know for glucose to be absorbed, we need insulin, right? From blood to cells. But glucose to be absorbed, we need insulin. But is that always the case? No, it's not. Because for fructose, we don't need it. Insulin is not required for the entry of glucose into the eyes, neurons, and the kidney. So large amounts of glucose can enter these cells during hyperglycemia, increased glucose. So what this means is for most of your body, we need glucose and insulin for being absorbed. But in the three specific areas, eyes, neurons, and kidneys, we don't need insulin. Insulin is not required. So large amounts of glucose enters these cells during hyperglycemia, increased glucose, because we don't have insulin, right? So I need you guys to remember eyes, neurons, and kidney, because this is where large amounts of glucose can be accumulated, okay? Therefore, loss of glucose, right, will turn into sorbitol, which starts to accumulate. Remember the whole pathway right here? Glucose, sorbitol, fructose. In eyes, kidneys, and neurons, glucose can enter without insulin. And then when you have a state of hyperglycemia, a lot of glucose can enter these areas because insulin is not required. And then this will start to accumulate. And in the eyes, kidneys, and neurons, it can be turned into sorbitol. Now, the funny thing is, in eyes, kidneys, and neurons, we don't have sorbitol dehydrogenase, right? That sorbitol dehydrogenase, the enzyme is pretty low for eyes, neurons, and kidneys. So what does that mean? If we don't have sorbitol dehydrogenase, this will start to accumulate. Fructose is not being made. And if this accumulates, that's very bad. So sorbitol, when this accumulates in the eyes, kidneys, and neurons, there's a lot of multiple stuff that can happen. Now, sorbitol accumulation will cause swelling leading to cataract formation and nephropathy. For cataract, if you guys don't know, something with the eyes, eye with cataract. If you guys read this in the exam, remember this picture. And why is that happening? Because we don't have sorbitol dehydrogenase in eyes, kidneys, and neurons, right? Kidneys, nephropathy, and eyes, we have cataract. So remember, sorbitol accumulation, because we have very low sorbitol dehydrogenase, no fructose is being made, this is being accumulated. What will happen? Cataract. Okay, that's about it. If this is hard, please tell me, guys. Please ask me questions. Yeah, but it's kind of straightforward as well. Just a lot of information at the same time. Okay. Now we're going to be moving on to the main part of this lecture, which is fructose metabolism. When you look at this, you will get overwhelmed because a lot is going on. We have this pathway, this enzyme, two products, a lot. But we're going to go in simpler terms, okay? So there's two pathways that fructose can take to be broken down, right? We have the low affinity pathway, the high affinity pathway. Low affinity pathway right here. For the low affinity pathway, we have the enzyme hexokinase. You guys know this already from glycolysis. Again, it can also be used here to break down fructose and to fructose 6-phosphate. This is low affinity. So majority of the times we're going to use fructokinase, not hexokinase, okay? So for fructose 6-phosphate, we already know the PFK1. Just a little recall, PFK1 was the rate-limiting enzyme for glycolysis. Fructose 1,6-bisphosphate, we know glycolysis is going to continue the glycolytic pathway. Low affinity hexokinase, just keep that in mind. Now, the high affinity we have, which is fructokinase, right here, this is high affinity. Remember, fructokinase also needs ATP to be broken down into ADP. So fructose will be broken down to fructose 1-phosphate, not 6-phosphate, fructose 1-phosphate through the help of fructokinase and ATP as well, making ADP. Okay? Then fructose 1-phosphate will use an enzyme called aldolase B, not aldolase reductase where we took before, aldolase B. Okay? Aldolase B, which is the rate-limiting enzyme of fructose metabolism. Okay? Fructose metabolism by the help of fructokinase, right? So aldolase B, that will make DHAP, say DAP or whatever, and then glyceraldehyde. This can also enter glycolysis. So this DAP can be used for fatty acid esterification. Just keep that in mind. But the whole purpose of fructose metabolism is DHAP or glyceraldehyde, which can go into glycolysis. Now let's make this more simpler. If this was difficult. Now, Fructose metabolism is almost always metabolized in the liver. So most of the time it's going to be in the liver. Fructose can either take a major, remember when I said major pathway, high affinity pathway, which is the fructokinase pathway, major fructokinase, fructose 1-phosphate, or minor, minor, which was low affinity hexokinase pathway. Most of the time, it's going to take this pathway. Hexokinase has a low affinity for fructose, as we said, hence most of the times fructose will take the major pathway in the liver, right? And okay, now it gets a little hard. What I mean by aldolase B, which was again the rate limiting enzymes for fructose metabolism, aldolase B has less affinity for fructose 1-phosphate than for fructose 1,6-bisphosphate, making aldolase B the rate limiting enzyme for fructose metabolism. Keep this point in mind. Aldolase B has less affinity for fructose 1-phosphate than fructose 1,6-bisphosphate. Aldolase B has less affinity for fructose 1-phosphate and has more affinity for fructose 1,6-bisphosphate. That's why this is the rate limiting enzyme. Now, fructose 6-phosphate can be converted into glucosamine 6-phosphate, which is the precursor of all other amino sugars. This is a very important point that the doctor mentioned in your slides. What he meant to say is that, as you guys can see, that you have fructose 6-phosphate, whatever. Now, fructose 6-phosphate, right here, this fructose 6-phosphate can be converted into glucosamine 6-phosphate. Now, glucose 6-amine phosphate can be converted into proteoglycans, glycolipids, glycoproteins. I mentioned it here as well from the slides. Fructose 6-phosphate can be converted into glucosamine 6-phosphate, which is the precursor of all other amino sugars. So glucosamine, other amino sugars that could be glycolipids, proteoglycans, and glycoproteins. Just keep this in mind. I highly doubt they will ask that, but they could. They might. So again, fructose 6-phosphate can be converted into glucosamine 6. What was it? Glucosamine 6-phosphate, and then that can be converted into protein. There's a little difficulty. Yeah, right here. Now, fructose 6-phosphate can be converted into glucosamine for proteoglycans, glycolipids, and glycoproteins. Okay. And these, just keep this in mind, these three circled in red. Okay. Now, this is also very, very, very high yield. It's bypass of fructose and glycolysis. Now, what do I mean by this? What do I mean is that when fructose is being broken down by fructokinase into fructose 1-phosphate, and then aldolase B is breaking down fructose 1-phosphate into DHAP or glyceraldehyde, these two can go back into glycolysis as fructose 1,6-bisphosphate or glyceraldehyde 3-phosphate. What they mean by bypass is that it bypassed the PFK1, which was the rate limiting enzyme, okay, which are activated by insulin. So let's say you don't have a lot of insulin, there's a problem, whatever, and we need glycolysis to produce ATP. They can use fructose metabolism to be broken down by fructokinase, aldolase B, back into DHAP and glyceraldehyde right here to be going back into glycolytic pathway, which is fructose 1,6-bisphosphate and glyceraldehyde 3-phosphate, without using the PFK1, which is stimulated by insulin when we don't have insulin. In diabetes, now this is also very important. In diabetes, hepatic gluconeogenesis is increased due to insulin deficiency. Now, fructose metabolism right here provides substrates for gluconeogenesis. So in diabetes, insulin is not being produced a lot. Fructose metabolism provides the substrates, which is fructose 1,6-bisphosphate and glyceraldehyde 2-phosphate by the enzyme aldolase B. So it basically bypasses the steps of hexokinase and PFK1. Both hexokinase and PFK1 are both stimulated by insulin, but in diabetes, we don't have a lot of insulin, so we're going to bypass by fructose metabolism. Please remember that. Okay, fructose metabolism can bypass in glycolysis. That's all I need you guys to remember. What enzymes again? Aldolase B, and in diabetes, it also helps fructose metabolism provide substrates right here. These are the substrates: fructose 1,6-bisphosphate and glyceraldehyde 3-phosphate into a glycolytic pathway. Okay, any questions, guys? Please feel free to ask me. Okay, now we're going to be speaking about the high fructose diet when you eat a lot of fruits or whatever. So fructose 1-phosphate, is much faster than fructose 1-phosphate to glyceraldehyde, as aldolase B is a rate limiting step. Okay, let me just. Fructose 1-phosphate is much faster to glyceraldehyde because aldolase B is a rate limiting enzyme. Right? This is much faster than fructose 1-phosphate glyceraldehyde. Fructose to fructose 1-phosphate, we're using an enzyme called fructokinase. But for fructose 1-phosphate to glyceraldehyde, we're using aldolase B, which is a rate limiting step. Therefore, in high fructose diets, we have lots of fructose 1-phosphate is made, which then accumulates due to the slow action of aldolase B. This is being accumulated in high fructose diets, not glyceraldehyde, fructose 1-phosphate, because this is slow. This rate limiting step is slow. Now, fructose 1-phosphate, which is done by fructokinase, requires ATP to turn into ADP. I'll show you guys again. I mentioned this, but fructokinase, as you guys can see, it requires ATP being turned into ADP, right? Requires ATP to turn into ADP. When this reaction increases, ADP and AMP will accumulate, okay? When a lot of fructose 1-phosphate is being made, a lot of ADP and AMP is also being made. Now, when a lot of ADP and AMP will accumulate, there will be tend to breaking down by metabolism of ADP and AMP produces uric acid, hyperuricemia, which could lead to gout. This is a lot. Let's go slower. When fructose or fructose 1-phosphate in high fructose diets requires ATP to turn into ADP. When this reaction increases, if this reaction increases, then ADP and AMP will also accumulate. Now, what happens is when ADP and AMP will accumulate, ADP and AMP will be broken down, produces uric acid, hyperuricemia, which could lead to gout. If you guys don't know what gout is, just remember this picture. Literally, uric acid crystals. That's remember. A scenario came to you guys in the exam. They're gonna say this child or whatever was eating a lot of high fructose diet foods like fruits or whatever, and then he says increased uric acid hyperuricemia, which can then lead to gout due to all being fructose 1-phosphate accumulation, and then this ADP and AMP will accumulate. Okay, that was a lot, guys. If you guys have any questions, please let me know, because I'll be moving on into disorders of fructose metabolism. Okay. So, okay, let's just move on. Now, disorders of fructose metabolism, if fructokinase is deficient, we have essential fructosuria. If we have aldolase B, which is a rate-limiting enzyme for fructose metabolism, if this is deficient, we have hereditary fructose intolerance. Please keep this in mind. I have a table as well for essential fructosuria and hereditary fructose intolerance. Not much to understand, just mostly memorizing the names, but the symptoms are important as well. Essential fructosuria, which is the fructokinase deficiency, known as fructose can be used, and fructose in urine. This is so important because they're going to give you guys this as a hint in the exam question. They're going to say this person came to you presented with this, that, but then they're also going to say that fructose in urine was present, was seen. This will give you a hint or will lead you to essential fructosuria, right? You won't see that in hereditary fructose intolerance. You won't. Okay, that's what you can differentiate between this and that. So for hereditary fructose intolerance, we have aldolase B deficiency. Okay, aldolase B deficiency. We have severe fructose is trapped by phosphate and hypoglycemia and hyperuricemia, hepatic, and renal dysfunction. Guys, if we don't have aldolase B, right? If we don't have aldolase B, this will accumulate, right? If this is going to accumulate, more ADP will be used, right? And then if we broke down ADP and AMP, it will lead to uric acid, right? So hyperuricemia as well, and hypoglycemia as well, because glyceraldehyde is not being made, which can enter the glycolytic pathway, so no glucose is being made. So glucose is low, hyper, and this ATP is accumulated. This is accumulated, so then this will lead to hyperuricemia, which can lead to gout as well. And we also have hepatic and renal dysfunction. But just remember, fructose in urine and hyperuricemia, hypoglycemia for a hereditary, essential fructosuria for fructose in urine. And remember the enzyme deficiency. Okay. Oh, just before we end fructose, we also have mannose to fructose 6-phosphate. You guys already know about hexokinase. Don't keep this in mind. What I need you guys to keep in mind is phosphomannose isomerase from mannose 6-phosphate into fructose 6-phosphate. This enzyme, phosphomannose isomerase, is used. Please keep this in mind. And that's why mannose is not essential in diets, by the way, because we already is being made from fructose 6-phosphate, right? So just know that mannose is converted into mannose 6-phosphate by hexokinase. We already know that. And then this is an important part: mannose 6-phosphate into fructose 6-phosphate by phosphomannose isomerase. Okay. This is the most important enzyme in mannose. So whenever you think of mannose, think of phosphomannose isomerase. It's in the name. Okay. And the most intracellular mannose is synthesized from fructose, not essential to diets because fructose can be made and then mannose can be made. Very low yield, but they asked us a question, so they might ask you guys a question in your final as well. So remember, it's not essential in diet because it can be made from fructose 6-phosphate. Okay. Let's move on. Okay, checkpoint. If you guys have any questions, please let me know. And if you guys don't have any questions, I have a question for you. Okay. So let's do the question. How does fructose metabolism bypass regulation by insulin? Okay. So you said B? Yes, it is B. Nice. I'll explain it as well. Let's go through all the questions. Oh, and by the way, a little tip for you guys in the exam: whenever you solve MOL practice questions or past questions, use elimination method because most of the other choices they don't seem right, like in the question. So you guys could have a better chance of getting the answer right. So A, fructose is directly taken up by insulin-dependent glutamate transporters. We literally said fructose is independent of insulin. It does not need insulin. It does not, no, insulin independent, no. Then we also have fructose enters glycolysis downstream, the regulatory step catalyzed by PFK1. It does not, it literally bypasses PFK1. So it literally enters glycolysis without using PFK1, which is the rate limiting enzyme of glycolysis. And then you have fructose. It's stored directly as glycogen without insulin regulation. I don't even know what this means. Fructose does not require conversion to intermediates for glycolysis. Doesn't make sense as well. So B is the one that makes most sense, and it's correct. Okay, let's move on to the next question. Okay, this is a lot. Okay, I'll read it. A child born and raised in Chicago plans to spend the summer on a relative's fruit farm and help with the harvest. The summer passed uneventfully, but several days after the harvest began, the child became jaundiced and very sick. On admission, yeah, it is B. Nice. Yeah. Inshallah. Okay. I'll still read it for completion's sake. On admission to the hospital, the following clinical findings were recorded in addition to expected hyperbilirubinemia. The patient was hypoglycemic, had a markedly elevated rise in blood fructose concentration, and was hyperlactic. So we know blood fructose concentration is very high. Further history revealed that during the harvest, it was customary for the family to indulge in fruit-filled meals. So what this means is that they have a high fructose diet and to snack freely on fruit while carrying out the harvest. This elevated blood fructose in the child was more likely due to which of the following? It is B, defective hepatic aldolase B. I'll show you guys. Let's just go back. High fructose diet as well. Look, high fructose diet, we also aldolase B is deficient. Fructose 1 phosphate will accumulate, and that's about it. Pretty simple. Does it keep on? Okay. As you guys can see, your fructose, the fructose 1 phosphate is much faster than fructose 1 phosphate to glyceraldehyde. So this is being accumulated because this is much faster than aldolase B because that's the rate limiting step. Whenever you have high fructose diets, fructose 1 phosphate is accumulated because of aldolase B. So whenever you guys see high fructose diets, remember aldolase B is slow. That's why it's being accumulated. Okay, nice. Okay, we're done with the first objective, which is the fructose part, and now we're going to be speaking about galactose. So, alhamdulillah, we're done with the first high yield major part of the lecture. This is also high yield, but it's shorter. No, no, we did this. We didn't skip a question. There's that. That was it. This was the first question, second question, right? And then galactose. Yeah, no, it's okay. Okay. Now we're going to be speaking about galactose. Very important as well. Shorter than fructose, alhamdulillah, but also very important. Okay. A little overview of galactose. Now, we already know a major source of galactose is lactose. Lactose is the disaccharide, galactose is the monosaccharide. Disaccharide of glucose and galactose and obtained from milk. Pretty simple background. Lactose and milk, we already know this. Main site is also the liver. Remember this for galactose and lactose. It is synthesized from glucose in large quantities and actively lactating mammary gland. I need you guys to also remember this part, lactating mammary gland. That is where lactose is being made from lactose. And again, enters in the formation of lactose, glycolipids, glycoproteins, and proteoglycans. And this is just telling you that there's a lot of ways galactose can enter. In other forms could be lactose, could be glycolipids, could be glycoproteins, and could be proteoglycans. It's pretty simple. Yeah, this is very simple. Let's move on. Okay, so now we have galactose metabolism. Again, I know this looks a lot and very like what the hell, but it's very easy. Inshallah, it'll be very easy. Let's go step by step. You guys need to know it has 3 roots, right? And that's about it. Okay. Now, glucose can be converted into galactose. Thus, galactose is not essential in that diet. What was also not essential in the diet? Mannose. Remember, who said mannose is not essential in the diet because it could be made from fructose 6 phosphate. Again, same thing here. Galactose is not essential in the diet. It can be converted by glucose and many other substances. Right now, let's do the first route. Galactose to galactitol is done by aldolase reductase. Okay, aldolase reductase. We also had aldolase B in fructose metabolism, if you guys remember. So this is different. The second part, aldolase reductase and aldolase B. Okay, now we also have galactose into galactose 1 phosphate by galactokinase. Very, very important. Galactose 1 phosphate can be converted into UDP galactose. Okay, and the UDP galactose can be converted into UDP glucose by the enzyme UDP4 epimerase. Please remember that UDP4 epimerase is very important, and galactokinase. And galactose 1 phosphate can be converted into glucose 1 phosphate by UDP uridyltransferase. The most important enzyme you guys need to know in galactose metabolism, right here. You have UDP uridyltransferase. And glucose 1 phosphate can be converted into glucose 6 phosphate by phosphoglucosamidase. Again, just remember that. Now, most important deficiency in galactose metabolism is galactose 1 phosphate uridyltransferase right here. UDP uridyltransferase from galactose 1 phosphate into glucose 1 phosphate. This is the most common deficiency. Okay, and galactokinase is also important to know. I mentioned that. So if I had to choose 2 enzymes to remember from this slide, I would say galactokinase and UDP uridyltransferase. But UDP uridyltransferase way more than galactokinase. But I suggest to remember all the enzymes. Okay. Let's move on. Okay. Again, we're going to be speaking about disorders of the galactose, which is called galactosemias. Galactosemias. Whenever you see galactosemias, just remember they're disorders of galactose metabolism. So either galactokinase has gone wrong, galactokinase deficiency, no galactose 1 phosphate is being produced, and then no glucose 1 phosphate. This whole thing is not being produced just because of one enzyme, galactose kinase. Or we have also classic galactosemia, which is the most common deficient enzyme, UDP uridotransferase. Remember, the most common deficiency we need to know is UDP uridotransferase, okay? And yeah, that's it. And also UDP epimerase, 4-epimerase, phosphoglucomutase, but classic galactosemia, which is the most common one, is done by UDP uridotransferase, and then galactokinase, which is galactokinase deficiency, right? That's about it. So let's move on because we're going to be revisiting this concept. So galactosemias, which is the disorder of galactose metabolism. The definition of galactosemias is basically increase in blood galactose concentration due to metabolism dysfunction. It could be due to this enzyme, due to this enzyme, due to this enzyme, a lot of enzymes, but mainly these two, right? UDP uridotransferase and galactokinase. Now, that leads to an increase in blood galactose concentration. Now, inherited defects in galactokinase or uridotransferase, which is the most common, or 4-epimerase. But please keep in mind, uridotransferase, UDP uridotransferase is the most common one. Or 4-epimerase. Now, can cause many effects. Cataract. Remember we mentioned cataract and fructose being made and sorbitol accumulation. Again, galactosemias is also another possible way of cataract being formed. By galactitol accumulates due to aldolase reductase. This is being accumulated, which was leading to cataract due to this enzyme, aldolase reductase. Liver failure, mental retardation, and galactosuria, exertion of galactose in the urine. Okay. And I need you guys to remember number 1 and number 4. Cataract. Galactitol accumulates due to aldolase reductase, and galactosuria, exertion of galactose in the urine. Okay. These are the most common effects. Please keep this in mind and for galactosemias, okay? Okay. This is the most important table you guys need to know. Galactosemias, we have 3 inborn errors of related to galactose metabolism. So type 1, type 2, type 3. Type 1, which is the most common one, UDP uridotransferase, which is galactose 1 phosphate uridotransferase, most common. Accumulating metabolites, we're going to have galactose, we're going to have galactitol, we're going to have galactose 1 phosphate, we're going to have galactonate. Guys, galactonate is new. Remember that, galactonate. And galactonate is only found in UDP uridotransferase. That's a way to differentiate between these 3 types of enzyme deficiency. As soon as you guys see galactonate, you're gonna think of UDP uridotransferase being deficient. Okay. Then you have galactokinase, which is rare. You're gonna have galactose and galactitol being accumulated, and then you're gonna have UDP galactose epimerase. Galactose 1 phosphate and UDP galactose. Okay, so the main one is UDP uridotransferase. Okay, now galactonate is the oxidized form of galactose produced by galactose oxidase, and only found in deficiency of galactose 1 phosphate UDP uridotransferase, not galactokinase or galactoepimerase. Okay, galactokinase deficiency causes galactosuria, galactose in the urine. Okay. Okay. Now, Now I need you guys to remember differentiating points between type 1 enzyme deficient and type 2 enzyme deficient. So for type 1, we have galactonate being accumulated. Okay. And for galactokinase, we have galactosuria, which is galactose in urine being accumulated. Okay. Now, this is the differentiating point. As soon as you see galactosuria, which is galactose in urine, you're gonna think of galactokinase being deficient. As soon as you guys see galactoconate or a lot of accumulating metabolites, we're gonna think of UDP uridotransferase. Okay. I hope that's simple for you guys. And just keep this table in mind whenever you're solving questions, and you're gonna get almost every single question right, inshallah. If not every question. Okay, checkpoint for galactose. If you guys have any questions, please let me know. And if you guys don't, I have a question for you guys. A patient with galactosemia presents with increased galactose and galactitol levels, but normal levels of galactose 1 phosphate. What is the likely enzyme deficiency in this patient? So we have galactokinase, galactose 1 phosphate. So as soon as you guys see normal levels of galactose 1 phosphate and increased galactose and galactitol levels, but normal levels of galactose 1 phosphate. Normal levels of galactose 1 phosphate, normal levels. Okay. And then we have these two are increased. So it's going to be galactokinase. Okay, nice. You got it right. Yeah, galactokinase. I think that's the only question for galactokinase. If you guys want me to have more questions, or if you guys want to ask more questions, please let me know. Okay, we're done with our second objective as well. Now we're going to be doing our third objective, which is everything about lactose. If you guys have any questions, please let me know. And okay, so if you guys don't, let's begin. Lactose. Remember, we mentioned lactose is a disaccharide that yeah, you guys, you can ask a question if just type it in chat. I think soon. I think by tomorrow, inshallah, it might be uploaded. So don't worry. Okay. Yeah. I hope that answered your question. Okay. They don't include first lectures? No. These PAL sessions include only the post midterm lectures. We already did the other PAL sessions for pre-midterm. So yeah. Yeah. Okay, let's continue with lactose now. Okay. Now, lactose is a disaccharide that consists of galactose and glucose. We already know that. Remember, I need you guys to remember because it came as a question that lactose is known as a milk sugar, which is produced by mammary glands. Remember when I said keep that in mind? Yeah. Now that is where you remember this. And lactose is known as milk sugar. Okay. Lactose is known as milk sugar and produced by mammary glands. Okay. And we have uh, synthesized by lactose synthase and Golgi apparatus. Please remember this, lactose synthase. This is what it's uh, synthesized by. This enzyme is very, very important. Thank you. Lactose synthase is made up of two proteins, A and B. Okay, guys, it gets a little confusing here, but please focus. Lactose synthase is made up of two proteins, A and B. Where it is made? In Golgi apparatus. Okay. Now, protein A, which is the first lactose synthase A, is found in a lot of body tissues, but only protein B is found in only lactating mammary glands. Okay, that is where it is known as a milk sugar, and only protein B. They might ask you a question about the differentiation. Protein A is found in a lot of body tissues, while protein B is found in only lactating mammary glands. Okay, and now you have lactose synthesis in mammary glands, which is protein B, is stimulated by prolactin hormone. Very, very important. Prolactin hormone is the hormone that stimulates lactose synthesis in mammary glands, protein B. This hormone is very important. Please keep this in mind because I'm pretty sure they're going to ask you a question about it. Just by this one point, prolactin hormone. Okay, let's continue. Okay. Now we have lactose intolerance. Please also another high yield point about lactose intolerance. It's also called lactose deficiency or hypolactasia, is the inability to digest and metabolize lactose found in milk. So the enzyme lactase is being deficient here in lactose intolerance. Okay. And common symptoms, please remember these common symptoms because whenever they ask you a question about lactose intolerance, they ask you, they literally mention these symptoms: diarrhea, nausea, and abdominal bloating. These three always come together for lactose intolerance. And they probably will ask you a question about lactose intolerance. Okay. And it's why does it happen? Because of the enzyme lactase being deficient. Okay. Uh, any questions here? If not, we can proceed. That's literally it about lactose. No more info about lactose. That's literally it. And there's a question for you guys to solve. You were examining a patient complaining of cramping in the lower belly, bloating, abdominal bloating, gas, and diarrhea. And these symptoms appear within 30 minutes to 2 hours after the ingestion of dairy products. They also mention dairy, so something like milk, whatever, dairy product. You advise your patient to avoid foods that contain a particular sugar. Which of the following is a sugar this patient should avoid? Glucose, no. Lactose, yes. And it is B. Thank you. Yes, and the others don't make sense. And I think that's about it. Yes, and we're done. Thank you guys for watching this PAL session. If you guys could scan this QR code for the feedback form, that would be amazing. And good luck, guys. Inshallah, you guys will do well. And salam alaikum.